Hello friends, so we are going to discuss about cancer genes. So the first one is proto-oncogenes. So these genes are normally responsible for growth and proliferation, but if there is gain of function in proto-oncogenes, then can become oncogene and there will be cancer. Here these are the two normal alleles. If there is gain of function in one of the allele, that can create heterozygous state and it can be oncogene. Okay, a single copy involvement is enough to cause cancer. Now here we have shown the rough diagram of growth receptor and growth factor. So if growth factor binding to growth factor receptor, then it will activate protein A that can cause activation of different next proteins, and that can cause activation of transcription factors ultimately, and this transcription factor increase transcription of cancer causing genes including your cell cycle regulator like cyclins and CDKs. Clear? We will discuss in detail in separate video about these all receptors. The second one is your tumor suppressor genes. So they are normally responsible for put a break on cell growth and proliferation. So if there is loss of function in this type of genes, that can cause cancer. So these are the normal alleles. Suppose if there is loss of function in one of the allele, that is a heterozygous condition is created, and this heterozygous condition will increase the risk of cancer. Clear? So this is about tumor suppressor genes. Now if both alleles are affected, there will be homozygous condition, and in this condition there will be cancer. Clear? And this is the basis of Knudson two-hit hypothesis. Now the third one is apoptosis related genes. So there are two types of genes, pro-apoptotic, anti-apoptotic. So pro-apoptotics, that is if there is loss of function, is pro-apoptosis, means there will be no apoptosis, so there will be increased chances of cancer. If there is gain of function, anti-apoptotic, means there will be anti-apoptosis, means there will be no apoptosis, that can also increase chances of cancer. Clear? And one of the example of anti-apoptotic gene is BCL2, which is seen in follicular lymphoma. Okay, so if there is gain of function in BCL2, that can cause follicular lymphoma. The third one is your DNA repair genes. So if there is loss of function in DNA repair genes, then there will be accumulation of mistakes in DNA, and that can cause cancer. There are three types of genes we have shown here. Mismatch repair genes. Okay, and if there is mutation in this mismatch repair genes, means if there is loss of function in mismatch repair genes, that can cause your hereditary non-polyposis colon cancer or Lynch syndrome. Clear? HNPCC or Lynch syndrome. The second one is if there is loss of function in nucleotide excision repair genes, that can cause your xeroderma pigmentosa. If there is loss of function in homologous recombination repair genes, that can cause Fanconi anemia, Bloom syndrome, ataxia, and BRCA associated breast and ovarian cancer. So this is the fourth DNA repair genes, which include mismatch repair genes, nucleotide excision repair genes, and homologous recombination repair genes. The fifth one is your tumor host cell interactions, certain genes which are involved in tumor microenvironment also. We will discuss about each of them in different video in detail. Now the second part is genetic changes which are causing cancer. The first one is point mutations. So there will be point mutations are the change in sequence of DNA at a single position. Okay, and that can also cause your cancer, and it can be assessed by PCR sequencing. Now there are two types of point mutation. It might be activating or inactivating. Activating means if there is gain of function. If there is activating mutation in proto-oncogene, means if there is gain of function in proto-oncogene, then there will increase protein expression of those genes which can cause cancer. Okay. If there is inactivating mutation, means loss of function in tumor suppressor genes, means decreased protein expression, which is responsible for tumor suppression, and that can also cause cancer. And both of these are assessed by immunohistochemistry. Clear? Here shown, this is your antibody, okay, against tumor protein. This is a target protein of tumor cell and chromosome attached with the antibody. Clear? Okay, it is responsible for color change when antibody will react with antigen. Okay, this is the basis of immunohistochemistry. In cell, if brown color is appearing, then positive; if not brown, then negative. Now the second one after point mutation is your gene rearrangement. Gene rearrangement involves either translocation or inversion. Translocation means between two chromosome, and inversion means on same chromosome. Clear? And both can be assessed by karyotyping or fluorescent in situ hybridization, and FISH is more preferred in compared to karyotyping. Now both can cause change in the locality of genes. Okay, not the actual sequences. These both cause a change in locality of gene, not the actual sequence. Clear? And this will lead to certain rearrangement. Okay, now consequence of rearrangement are two possibility: either this gene 1 and gene 2 combined, okay, and their mRNA would be made together as a single sequence, that is the new fusion mRNA transcript, and this new mRNA will make a new protein, chimeric or fusion protein, and that can increase or decrease function as before, and that can be responsible for cancer. Clear? So this is the possibility number 1. The possibility number 2 is, so gene 1 and gene 2. Gene 1 will be proto-oncogene and gene 2 will be promoter sequence. It will promote the gene 1. Okay, it will promote the transcription of gene 1, increase transcription, increase expression of proteins. It can be also assessed by IHC. And possibility number 2 is seen more in lymphoma, and possibility number 1 is seen more in leukemia. Okay? The third one is your deletion. Okay, deletion means loss of functions, loss of any nucleotide sequence. There will be loss of function. That loss of function means loss of protein. That can also cause your cancer. The fourth one is amplification. So we have discussed the first one is point mutation, then we have discussed rearrangement, then deletion, then amplification. Amplification means increase gene copies. Okay, that will cause increased protein expression. Okay, so these are the four methods. The fifth one is aneuploidy, means chromosome will not be in multiple of 23. It can also be assessed by karyotyping. Now coming to the one more thing, fluorescent in situ hybridization. Fluorescent means level nucleotide to make DNA probe. In situ means in its place, no extraction of DNA from its place. Hybridization means complementary base pairing concept. Yeah. So this is about your genetic causes. Clear? Now one more point remaining, that is miRNA. Okay, this is responsible for silencing of target genes, and they are the example of non-coding regulatory RNA, as we have discussed in our previous video. So if there is gain of function in those miRNA which target TSG, tumor suppressor gene, then cancer will be there. If there is loss of function in those miRNA which target your proto-oncogene, then there will be cancer. Okay, so these are the main genetic causes, that is point mutation, deletion, amplification, rearrangement, aneuploidy, and this miRNA. Clear? Now coming to the hallmarks of cancer. So the first hallmark is self-sufficiency in growth signals. Okay, it does not require growth signals, it proliferates on its own. So this is the first hallmark. The second hallmark is their insensitivity to growth inhibitory signals. Okay, they do not listen to breaks in proliferation, it continues to proliferate. The first one is self-sufficiency, the second one is insensitivity, the third one is altered cellular metabolism, that is also coming under Warburg effect. Cancer cells are aerobic glycolysis and not pure mitochondrial oxidative phosphorylation. Okay, make less ATP but gives a lot of carbon rich intermediate to make new cell component, that is known as Warburg effect. The fourth one is evasion of apoptosis, and mainly intrinsic pathway evasion is more important. Okay, so if there is DNA damage, that will be sensed by your sensor proteins. Okay, and that can signal to, that can see activating signal to pro-apoptotic genes. Okay, and negative signal to anti-apoptotic. This is the general way, this is the normal way. Okay, normal way of intrinsic pathway, that if there is DNA damage, then it will be sensed by sensor proteins and it will say message to your pro-apoptotic genes. Okay, and these pro-apoptotic genes will increase mitochondrial permeability, leading to release of cytochrome C. Okay, and cytochrome C will bind to APF and cause activation of caspase 9, and that will cause activation of educational caspases, and that can cause apoptosis, and it inhibits anti-apoptotic. But if there is loss of function in pro-apoptotic, that can cause cancer. So loss of function of pro-apoptotic gene is seen in cancer. If there is loss in APF1, it can also cause cancer. So loss of this is seen in cancer. Mitochondrial permeability, increased mitochondrial permeability also lead to release of certain proteins, SMAC and DBLO, which are inhibitory to those proteins which inhibit apoptosis. Again, SMAC and DBLO are those proteins which are inhibitory to those proteins which try to inhibit apoptosis. Okay, so if there is increased level of these proteins, that can also cause cancer. The fourth one is your immortality, that is limitless replicative potential. Okay, normal cell 50-70 division, there will be shortening of telomeres. Okay, and there will be exposure of telomeric sequences. If p53 is functional, then it will detect the telomeric sequence and lead to senescence. But if there is p53 is non-functional, suppose if p53 is non-functional as in cancer cells, then there will be less exposure of telomeric sequences. And one more point here, telomerase will now come and add sequences. Okay, activation of telomerase will be there in cancer patient, which adds sequence to telomeres and then does protect the end of chromosome. Okay, and that can lead to cancer. So this is the pathway for your cancer cells, and this is for normal pathway. Clear? Now the fifth one is sustained angiogenesis, because there is rapidly growing tumors and it can cause hypoxia, and hypoxia can occur if less blood supply, because they are rapidly growing tumors, so they need more supply. So there will be hypoxia, and hypoxia will lead to either necrosis and angiogenesis. Okay, angiogenesis is stimulated by VEGF, and VEGF inhibitors are being used to deprive tumors of their new blood supply. Clear? So we can also use VEGF inhibitors in our treatment. So hallmarks of cancers are, the first one is it's limitless growth. Okay, sustained angiogenesis. Now the seventh point is your ability to invade and metastatic. Okay, that is a metastatic cascade, which we will discuss later on. Now coming to the proto-oncogenes again. Okay, so most common target for therapy are the proto-oncogenes, because they are active. Okay, and we try to inactivate these proto-oncogenes. So most common targets for therapy. So the first one is your growth factors and growth factor receptor. The first one is your EGFR family. Okay, and this is the family which has its own receptor tyrosine kinase. Two examples of this, ERBB1 and ERBB2. Clear? It is also simply called as EGFR, and ERBB2 is also called as HER2. Now this ERBB1 amplification is shown in your squamous cell lung carcinoma. Okay, if there is point mutation in ERBB1, that can cause adenocarcinoma of lung. Okay, and also can cause gliomas and colon cancer. Now amplification of ERBB2 is seen in your breast and gastric carcinoma. Now we have also drug which is targeting ERBB1 and ERBB2. So ERBB2 targeting drug is trastuzumab. Okay, and now the second one is your ALK, that is anaplastic lymphoma kinase. Okay, this is also a type of receptor tyrosine kinase, and there will be two types of changes in this, either mutation or rearrangement. So mutation in ALK lead to your neuroblastoma, hereditary hemorrhagic, okay, and telangiectasia type 2. The rearrangement in ALK, that is anaplastic lymphoma kinase, can lead to lung adenocarcinoma, anaplastic large cell lymphoma, and inflammatory myofibroblastic tumor. Clear? Now this is also a type of receptor tyrosine kinase. Clear? The third one is your PDGFR alpha or beta. It also has its own tyrosine kinase, and its mutation is seen in gastrointestinal stromal tumor, and its amplification and mutation is seen in glioblastoma. Okay? And if there is rearrangement in both alpha and beta, that can also cause myeloid lymphoid neoplasm with eosinophilia. Clear? The next one is your FMS like tyrosine kinase 3, FLT3. Okay, it undergo point mutation in acute myeloid leukemia. Okay, the next one is c-KIT. It is also receptor tyrosine kinase. Its point mutation is seen in gastrointestinal stromal tumor. Okay, seminomas, mastocytosis, and acute myeloid leukemia. Now the next one is JAK2 Janus kinase. It is a non-receptor tyrosine kinase receptor. Its point mutation is seen in polycythemia vera, essential thrombocytosis, and primary myelofibrosis. Clear? One is your B617F, means at 617th position valine changes to phenylalanine. Okay, and one inhibitor of the JAK2 is your ruxolitinib. Now the next one is ABL1. It is also example of non-receptor tyrosine kinase. Okay, and its gene is present on chromosome 9 and on chromosome 22 BCR gene is present. Okay, suppose if there is rearrangement in these two chromosomes, then two new chromosome will form, that new chromosome 9 and new chromosome number 22. Okay, and this new chromosome number 22 is contains of both ABL1 and BCR gene. So here there will be fusion of BCR and ABL1. Okay, it will form fusion protein. Okay, and it will cause overactivity of tyrosine kinase. Clear? And that can also lead to cancer. Now we can also give inhibitors like imatinib for this. Okay, now this new chromosome number 22 is known as Philadelphia chromosome. Clear? Now the next one is this fusion protein may be of three types: 210 kilodalton, 190 kilodalton, or 230 kilodalton. So they are specifically associated with different types of leukemia. So 210 is associated with chronic myeloid leukemia, 190 kD is associated with acute lymphoblastic leukemia, and this 230 is associated with myeloproliferative neoplasm with exacerbant neutrophilia. Clear? And this depends on types of BCR exon, because BCR is fusing with ABL1. Clear? That is non-receptor tyrosine kinase. ABL1 is example of non-receptor tyrosine kinase. Now moving to the next, that is signal transduction proteins. Clear? We have discussed about growth factor and receptor. Now coming to the signal transduction proteins. So the first one is your RAS. It is the most common proto-oncogene mutated in human cancers. Clear? Now RAS may be of many types such as KRAS, NRAS, HRAS. Okay, it is associated with pancreatic cancer, colon cancer, and lung adenocarcinoma. And most common genetic change in RAS is your point mutations. NRAS is associated with malignant melanoma, and HRAS is with bladder carcinoma. And they are the GTP binding proteins. Clear? Suppose if the growth factor binds to growth factor receptor, that will activate your tyrosine kinase activity, and that will convert inactive RAS to active RAS, and inactive RAS as GDP and active RAS as GTP. Clear? And conversion of GTP again to GDP will make active RAS into inactive RAS. And this active RAS set signal forward to MAPK and PI3K. Okay, and this will set signal to BRF, and this will set signal through AKT to mTOR. Now if suppose GTPase activity proteins, if GTPase activity will increase, then there will be increased conversion of GTP into GDP. Okay, and there will be increase in activation of RAS. Clear? Now GTPase activating proteins, one is your NF1. Okay, it is tumor suppressor genes, because it is inhibiting this pathway, so there will be no signaling pathway, so there will be no growth. So NF1, that is neurofibromin 1 gene. Okay, it is responsible for GTPase activating action here. If there is loss of function in NF1, then it will cause cancer in RAS cancer, because of the gain of function in RAS, because NF1 is inhibiting this pathway, and if NF1 is absent, then there will be no inhibition of this pathway. The next one is your PI3K. It is inhibited by P10 gene. Okay, if there is loss of function in P10, it can also cause gain of function in PI3K. There will be gain in function of PI3K, because PI3K is inhibited by P10 gene. Clear? And it is associated with Cowden syndrome. Now so this is about your signal transduction protein, RAS. Now the third one is your transcription factors such as MYC. So there are several types of MYC: NMYC, CMYC, LMYC, and they are associated with different types of cancers. Such as NY is associated with neuroblastoma, C with Burkitt lymphoma, and L with small cell carcinoma lung. Okay? And there will be amplification or overexpression of these transcription factors. Other transcription factors are MYB, JUN, FOS, REL, and this is involved in your Hodgkin's lymphoma.